Hey guys, welcome back. Here is Chapter 15, the chromosomal basis of inheritance. Chapter 14 was all about some theory and history about Sir Gregor Mendel and all his garden peas and what he discovered about the basics of inheritance and genetics, and then we applied that to what we know a little bit about humans and various human ailments and diseases and things of that nature. This chapter is more about the physical, what's going on inside the cell when those chromosomes are duplicating. So how can we take the theory and the ideas of genetic inheritance and apply it to what can be actually seen in a microscope? That's what we're going to do in Chapter 15. So keep in mind that back in Mendel's day, he didn't really know what chromosomes were. They didn't have the equipment, microscopes to actually see chromosomes, and it wasn't well understood how these factors or units of inheritance could be passed on from parent to offspring. Now, of course, we know a tremendous amount more than Gregor Mendel could have possibly imagined, and also Charles Darwin and his theories of evolution. Of course, he didn't have a very strong genetic component to his theories as well, because he didn't, even though he lived at the same time as Gregor Mendel, they didn't know each other, and they didn't communicate, and they didn't pass on their information to one another. So if they had done that, both of their ideas and theories would have been strengthened back in the 1800s, but it wasn't until probably the early 1900s into the 1920s where Gregor Mendel's work was kind of rediscovered, and it was really brought to light, these ideas of genetic inheritance. And now, of course, we can apply what we actually see in the microscope to those ideas. So Chapter 15 is what is going on inside the cell that corresponds with all that information that we just talked about in the previous chapter. So here's just a brief overview about which talks a little bit about locating the genes along the chromosomes. Now, of course, we know that genes are a portion of a big strand of DNA that makes up what we call a chromosome. But back in the 1800s, Mendel had no idea about this. He coined this idea or phrase of hereditary factors, but he didn't have a physical thing he could point to and say, and there they are. Now we can do that. So his hereditary factors were genes, but he didn't have the ability to see these genes. Now we can see them, we can locate them, we can even tag certain genes by tagging that particular portion of the chromosome with a fluorescent dye, and we can see the portion that affects perhaps one particular trait. Here's a cool photograph taken through a microscope, and you can see there's a couple of little highlighted areas. These little highlighted areas, these yellow dots, have been tagged with a fluorescent dye, and they mark the locus of a specific gene on the sister chromatids. There's a homologous pair of chromosomes here. This one and this one are homologous pairs, and those little dots are marking the specific location of a particular gene, and this would be during prophase of human chromosome development. So now not only can we see the chromosomes, but we have the ability to locate on that chromosome exactly where the gene is that controls a particular trait that we're talking about or studying. Pretty amazing. It would blow Mendel's mind. So starting with concept 15.1, it says Mendelian inheritance has its physical basis in the behavior of chromosomes. So now we have the ability to tie physical action within the cell with these ideas that Mendel first developed way back in the 1800s. Mitosis and meiosis were first described in the late 1800s, but like we said, these were just basically ideas. They couldn't physically point to where this was happening. The chromosome theory of inheritance states that Mendelian genes have specific loci or loci positions on chromosomes, and chromosomes undergo segregation and independent assortment. Remember, we talked about those two ideas that Mendel described called segregation and independent assortment, and now we know that the behavior of the chromosomes during meiosis can account for these ideas or these laws, and we can actually see it happen. So now we know what physical behavior kind of ties these ideas together so that we can see it, and this helps us understand that these factors that Mendel talked about that we now know are genes, they must be located on the physical chromosomes because we can watch this process happen now with the equipment that we have. So let's kind of step through a particular cross and kind of see what physically is happening that accounts for what Mendel saw in his offspring when he was dealing with his pea plants. So here we have the parent generation, the parental generation, the pea generation. The one parent is yellow, round seed. That's homozygous dominant for both traits. So it's a pure breeding. It would give nothing but yellow and nothing but round. And we breed that to another pea plant that is homozygous recessive. It gives nothing but the recessive gene or the recessive allele or a trait, green and wrinkled. So we cross those. They undergo meiosis, and then they produce gametes that only have the one allele that they can give. And then we're going to have fertilization, which is where those two are combined to create the first offspring. Then we have a process that we can look at to see the law of segregation and the law of independent assortment. And we'll kind of look at them individually, but this is just kind of an overview at the moment. So this will be the F1 generation. This will be the first generation that came from those parents that we just looked at. So here are the parents, a yellow round and a green wrinkled. Now we know that all of their children, offspring, are going to look yellow round. Even though they're heterozygous, so you can see right here, they have the green and wrinkled allele within them, but yellow and round is dominant, so all their children come out to be yellow and round. But we know that doesn't tell us their genetics. We look inside, they have both the yellow and the green, and the round and the wrinkled possible allele. Those plants are going to undergo meiosis to create their own gametes. And here's where we have our two laws. The law of segregation says the two alleles for each gene separate during gamete formation. So that means the large R for round and the little r for wrinkled, they're going to separate, and they're going to end up in different gametes. And the big Y for yellow, the small y for green, they're going to separate and end up in different gametes. And if you follow this on down the line, sure enough, you have some gametes that have the big Y, some that have the small y, some that have the large R, some that have the small r. On this side, we're looking at the law of independent assortment. It says alleles of genes on non-homologous chromosomes assort independently during gamete formation. And that just means that when the alleles line up in the center along the metaphase plate, they're going to do so randomly. So you could have one allele go left, one allele go right, and that's going to be completely random as to how that occurs. Now let's get to our F2 generation. This would be the grandchildren of the original parents. If we take a look at the F2 generation, we're going to find out some specific information about them. So if we look at their phenotype, what do they look like? Well, we're going to get a particular ratio of P's. We're going to see a 9-3-3-1 type of ratio. The vast majority of the P's are going to be yellow and round. Some of these could be homozygous dominant, some of them could be heterozygous for the trait, but that's what we're going to see. Then an equal number may be a mixture of the two traits. In other words, they're going to be dominant for one trait, recessive for the other. So here we have round green P's. They are dominant for the round, but they're recessive for the green. Here we have wrinkled yellow. They are recessive for color, dominant for color, but recessive for the wrinkled trait. And then we have this one little guy over here. He's homozygous recessive for both traits. So we have a 9-3-3-1 ratio in our phenotype of what we see. This is because of the two laws we just talked about. Mendel's law of segregation states that the allele pairs separate or segregate during gamete formation and randomly unite at fertilization. So the gametes that were formed will have a random possibility of being green or yellow, round or wrinkled. And then Mendel's law of independent assortment states that the allele pairs separate independently during the formation of gametes. That means that these traits are not tied together. In other words, you could be dominant in one trait and recessive in the other. Because of the way they line up and because of the fact that it is random, the way they line up with the metaphase plate, that means the traits are transmitted to the offspring independent of one another. So if the trait is on non-homologous chromosomes, or we also found out it could be on this homologous chromosome, but if it's far enough apart on that chromosome. But in this case, the law definitely applies if we're talking about non-homologous chromosomes, traits that are not on the same chromosome. Then those traits are going to be sorted randomly or independently. They're not going to be tied together. So here's our big overview. And basically we're saying we get what we see at the end here with these grandchildren, the F2 generation, for these two reasons. Number one, because the alleles will be segregated randomly into the various gametes. And number two, because the two traits we're looking at are not linked. They are sorted independently of one another. So this is what you're going to end up with because of these two laws, the law of segregation and the law of independent assortment. And this ties physically what's going on inside the cell with the idea that Mendel had, even though in his time he didn't have the ability to physically see it happening. So let's move on to some more modern experimentation that also helped to cement this idea that genes are located on specific chromosomes, and we can have physical evidence of this information. So the first solid evidence that associated a specific gene with a specific chromosome came from an embryologist, a type of biologist called Thomas Hunt Morgan. And Morgan is very well known because his experiments kind of centered around fruit flies. And fruit flies still today are one of the most used animals in a biology lab. Morgan's experiments with fruit flies basically provided really strong, convincing evidence that chromosomes are the location of these heritable factors that Mendel was talking about. Remember, prior to this, we didn't know physically where were these chromosomes. Well, we didn't call them chromosomes, but we physically didn't know where these heritable factors resided within the cell. And even though people knew chromosomes and had seen chromosomes, we still didn't have definite proof that that's where the genes, as we came to call them, were located. So Morgan's experiments helped to prove that. We've got a great video if you want to click on this link right here. I believe this is the video that Bill Nye hosts, and it's fairly lengthy, but if you want to go to the 342 mark, and that talks about Morgan and his fruit fly experiments. So why fruit flies? That seems like an odd choice. Well, there's a couple of good things about the fruit flies that made them really good test subjects for his particular experiments. So we're talking about Drosophila melanogaster. Hope I'm pronouncing that correctly. Drosophila melanogaster, a convenient organism for genetic studies. Why is it a good one? Well, number one, they breed like crazy. They produce a lot of offspring. So when you're doing genetic experiments, you want a lot of offspring so you can kind of move quickly through your experimentation. A generation can be bred every 2 weeks. Here we go again. Strong breeders every 2 weeks. Remember, you don't want to pick, say, a creature that is a large mammal that might take years before it develops into sexual maturity. So you wouldn't choose to study genetics by studying, say, elephants that might take years and years and years before they're sexually mature, or like humans, which might take at least 14, 15, 16, 18 years before the creature is sexually mature. You don't want a 20-year generation cycle. So 2 weeks, you can turn around generation after generation fairly quickly. And the last thing that makes the fruit flies really good for this type of experiment is they have very few chromosomes. They only have 4 pair chromosomes, which is great because that limits what you're having to look at and sort out. So they have 3 pair of autosomes and 1 set of sex chromosomes. And they work in the XXXY way that humans do as well. So that also makes it applicable, a little bit more applicable to us. So very few chromosomes, only 4 pair, 3 autosomes, and 1 set of sex chromosomes. So because Morgan was kind of a trailblazer, he often was the person who also came up with various ways of describing what he saw and vocabulary that we still use in genetics today. First, he noted what he called the wild type, or what he would call the normal phenotype. This is what you would see commonly in everyday fly population out in the wild. So, for example, one of the things he looked at was eye color. And red eyes is the wild type. That is what you normally see in fruit flies out there in the world, outside of the laboratory. So when he found a trait that was not the wild type, for example, we're going to see he had a fly with white eyes, he would call that the mutant phenotype, or the alternative phenotype. So Morgan worked for years in what came to be called the fly room at Columbia University. And over the years, he bred thousands of flies. And as a matter of fact, in his early days in his experiments, he sort of got frustrated because he wasn't getting the results he wanted. He wasn't seeing what he wanted. And he felt to some degree perhaps he had wasted his time breeding thousands of flies and not producing the scientific work that he wanted. But one day, he was viewing some of his fly specimens under the microscope. They would catch them live and they would sedate them, basically with a chemical, and they would put them under the microscope so they were still alive because they had to return them back to the vial for breeding experiments. One day he popped a fly underneath his microscope and he saw that it had white eyes. It was a male fly with very odd looking eye color, very different from the wild type that you normally see. So here on the left, this would be the wild type that you would normally see in a fruit fly, kind of this reddish orange eye. And this is what he saw in the microscope. He saw the first mutant fly that he had in his collection this white-eyed male fly on the right. So here's one of the experiments that Morgan became basically famous for. In one experiment, Morgan took his male mutant fly, the one with the white eyes, and he bred it with a female with red eyes, which would be the normal wild type. The first generation, the F1 generation, had all red eyes, which you would kind of expect. You would expect the red would be dominant and the white would be recessive. So that didn't surprise anybody, or didn't surprise Morgan. Then the F2 generation came about. In the F2 generation, he saw a 3-to-1 red-white eye ratio. Again, not surprising. That's what you would expect if you were to do a heterozygous for a trait. You would come phenotypically, you would expect in a Punnett square for 3 of them to display the phenotype of the dominant trait and 1 to display the recessive trait. But something he noted, only males had the white eyes. So he wasn't getting a random assortment of 3-to-1 with male and female both having the red and the white. Only the males had the white eyes. So what that helped Morgan determine was that the white-eyed mutant allele, the particular allele or gene that was different for that characteristic, must be located on the X chromosome. So Morgan's findings supported the chromosome theory of inheritance, that genes are located on specific locations physically on the chromosome. So let's break down his findings so we can understand this. So what he said was, because in the F2 generation only the males had white eyes, that mutant allele must be located on the X chromosome. Why did he think this? Why did he think the gene for eye color was related to sex? Well, number one, if it was not related to the sex chromosomes, then you would expect a 50-50 distribution of white-eyed flies, male and female. So just randomly, by chance, by probability, you have a 50-50 shot of having a male or female fly. So 50% of the white-eyed flies should be male, 50% should be female. But that's not what he saw. Only the males had white eyes. So that led him to believe, okay, we must narrow this down to the sex chromosomes. Remember, the sex chromosomes primarily control sex, but also they control other traits as well. So the allele for eye color must be located on the X sex chromosome. Why did he say that? Well, males would show the recessive phenotype, which again is the white eyes, with only one allele. Because remember, the males have an X and a Y, and there's no homologous allele on the Y chromosome to cover that mutant trait on the X. Females, for a female to show the white eyes, they would have to have been homozygous recessive, where both of their X's had the homozygous allele for white eyes. That, in his case, because of the breeding parents that he started with, would not happen. So he didn't see any white-eyed females. So again, he knew it was on the X chromosome, this mutant recessive allele, because in males, there was no allele on the Y chromosome to match up with this eye color, and so there was no way to overcome it. So the male shows recessive trait with only the one recessive allele. In the female, for her to show the recessive trait, she would have to have both recessive alleles on her 2 X chromosomes. So even if she had a recessive allele on one X chromosome, the red eye dominant allele on the other would cover that up and you wouldn't see it. So this is how he determined that this particular chromosome was located on this particular allele, this particular gene was located on this particular chromosome, on the X chromosome of the flies. Again, your book does a great job showing illustrations, so it's always good to look at an illustration. So this is kind of a drawing of what he did. So we had a red-eyed female and a white-eyed male. All of the offspring in the F1 generation had red eyes, which is what he expected. He expected this to be the dominant. So it didn't matter if they were male or female here. They all had red eyes. Then the next generation, he saw a 3-to-1 ratio, like you would expect if you did the Punnett square, but he noted that the sexes weren't equally distributed. So you notice that only the males here have white eyes. So there was no females with white eyes, and that is what caused him to begin to think, okay, this must have something to do with the X sex chromosome. and he was able to indicate that this trait, this gene for this trait, was located on that particular chromosome. So looking at the alleles that these flies passed, we're using W. The way that Morgan named his traits was after the first mutant that he saw. So the first mutant eye color he saw was the white eyes, so he used a W to represent this trait. And the plus represented the wild, without the plus represented the mutant. So in this case, we're looking at the trait for eye color, and he named it after the white. That was the first mutant he saw, so he used the W. So W plus is the dominant red eye trait, and W by itself, without the plus, is the mutant white. So the females here, they were homozygous dominant. She could only give alleles for homozygous dominant red eyes, W plus. The male, there was no allele on his little Y chromosome, so he only has the one allele for white that he can give. He can give the allele for the white recessive mutant trait, or nothing, based on that other particular chromosome, the Y chromosome. So then that tells you what the possibilities are for the F1 generation, and we pass that on down to the F2 generation. So if you look, the females, they could have homozygous dominant for red eyes, they could have the dominant trait and nothing, they could have the dominant trait and the recessive. Either way, all three of them were going to have the red eyes. But in the male, he could have the white recessive and the Y, because that's what indicates he's a male with the Y chromosome. With nothing to cover up this recessive trait, it's going to show. So the reason he has the white eyes is because he is male, and he has no homologous allele on the Y chromosome to help cover up that recessive allele on the X chromosome. So he's going to show the white eyes, and that would be seen, in this case, only in the males. So Morgan's work helped to prove that the chromosome theory of inheritance was correct, that specific genes are carried on specific chromosomes. And for his work, he won the Nobel Prize in Medicine and Physiology in 1933, and he is pretty darn well known throughout biology and the study of genetics. Thomas Hunt Morgan. Okay, the next concept, we're going to take a break and we'll come back to that. It'll be concept 15.2, and it talks about how sex or gender, male or female, that type of thing, is passed on to offspring. So it says sex-linked genes exhibit unique patterns of inheritance, and we'll talk about that in just a moment. Take a break, come back for concept 15.2.